Hi, everyone. My name is Mark Marisich. I'm Chief Strategic Officer at JAL Therapeutics. And I'm here today to talk to you about our platform of highly selective butyrylcholinesterase inhibitors to treat Alzheimer's disease, as well as other chronic and also neurodegenerative diseases. So the problem that we're addressing is that the old school class of cholinesterase inhibitors currently used to treat Alzheimer's disease inhibit acetylcholinesterase as an MOA. However, they all have serious side effects and poor efficacy since acetylcholinesterase is needed for brain function. So the solution that we bring is really highly selective inhibition of butyrylcholinesterase, or BChE for short. Since BChE is not required for brain function, yet elevated BChE is greatly associated with the disease. So our lead drug, JAL001, we're really focused on this and is designed to restore acetylcholine and cognitive function, addressing the symptomatic effects, and then also decrease the biomarkers associated with the disease progression in Alzheimer's disease modifying. So our business model is really focused on our novel small molecule drug platform and really looking at ourselves as a better, more effective frontline treatment for AD at earlier stages, replacing the old school cholinesterase inhibitors. So looking at the competitive river, you say, looking at us, we're far upstream from any of these other drugs out there that are in development. We're really focusing on highly selective BChE inhibition, and we're also addressing not only the symptomatic treatment, but also disease modifying as well. So looking at the market opportunity, really focusing on symptomatic, if you're looking at the TAM is 4 billion, the SAM is 3 billion, and the SOM is 2 billion. But if you were also to look at us as a disease modifying solution, the SOM could be 4 billion. And then on top of that, if we were also used as a prophylactic agent in conjunction in treatment with other chronic disease, it could be 6 billion. So looking at our financial model, really focusing on what we want to do right now is conduct the IND-enabling studies. We want to de-risk and add value. We've really, from the start, just focused on licensing with pharmaceutical companies or acquisition, but if it happens to be that we go IPO in the future, so be it. The deal size are typically around 1.5 billion for these types of targets. In terms of our executive team, founding roots at Cal State Long Beach in organic chemistry and also neuroscience. We have team members from pharma, Avenir Pharmaceuticals, as well as in commercialization and communications and life sciences. So in terms of our funding requests, we're looking for 2 to 5 million to help fund our IND-enabling studies. We're looking to file for IND in late 2026. Thank you very much.
Question, Mark. Can you talk about a little bit about the data, what you have to date on inhibiting APP, A-beta, tau, or any neuroinflammation?
Yeah, we have all that. We've conducted all sorts of studies, in vitro studies, ex vivo studies. On the APP, it was ex vivo with the human neuroblastoma cells. We decreased the amyloid precursor protein by about 47%, I believe. And then on top of that, the beta amyloid peptides, AB40, AB42, decreased those significantly as well. And then also on top of that, we are inhibiting the development of plaque in the hippocampus of transgenic mouse models of Alzheimer's. In fact, here is the little slide that shows that. If you can take a look at this, this is actually the transgenic mice. The images on the left for the immunohistochemistry, you can see the dark black blotchy, that's the plaque on the hippocampus. In the area highlighted by the green, those are the mice that are treated with our drug, and it's clearly inhibiting it, and it's across the board, it's statistically significant. So in addition to that, we've conducted the studies with the mice. There were no off-target effects in the mice across the board, and so it's doing what it's supposed to be doing.
Any impact on microglia?
Not that we are aware of at this point.
Thank you.
Yeah.
One question, actually, but I'll start with the first one. So you've listed a couple of companies in terms of what their timeline is and what their investments are. But obviously, for example, you've listed Anubis, which is a completely different MOA they're going after.
Yeah.
They're going to toxic proteins. So in terms of your kind of going forward plan that you have in terms of cost and what you need, regulatory path, are you comparing yourself to the companies that you've listed in your slide, or are you comparing more to, the current drugs that have been approved for the acetylcholinesterase? Which ones?
Yeah, we were just looking broadly across the board, drugs that were in either the Alzheimer's or CNS space, and this was just a reference. I mean, every deal is different and every situation is different. I mean, sure, we have Alzheimer's could be one potential indication. We have other indications that could be potential as well in the CNS space. And even broader than that, I mean, elevated butyrylcholinesterase is connected with many different things. Actually, if you look at the data, there's academic data saying that the elevated butyrylcholinesterase is directly connected with type 2 diabetes, and potentially that could be a treatment path for type 2 diabetes. So we have all these different indications covered under our IP. Again, we've been advised by our team at the California Life Sciences. We were part of the FAST program. They said, hey, just slow down. Just focus on one thing at a time. But this is the bigger picture that we have. If you look too at, for instance, type 2 diabetes, that's also directly connected with Alzheimer's. There's a greater increase, but we're just focusing on what we're doing for right now.
So I think that's actually fantastic advice, and I give you credit for focusing already on a very large, challenging, unmet need. To Farhana's point, right? So I mean, you showed that river of sort of like other companies in the space, and I'm glad you clarified. It was basically like whoever seems to be playing in the AD neurodegenerative space. But are you the only company, to the best of your knowledge, that is actually working on this particular MOA?
Yeah, we're, as far as we know, we're the only one who's addressing elevated butyrylcholinesterase. There have been other drugs out there, the cholinesterase inhibitors. They primarily focus on acetylcholinesterase, and they have inhibited a little bit of butyrylcholinesterase. However, we're super selective just for butyrylcholinesterase. We have a 68,000 fold selectivity. And the one thing about butyrylcholinesterase as well, it's, of course, not necessary for brain function. The purpose in the body is that of a toxin-fighter, but we're not trying to eliminate it completely. We're just trying to get it down to normal levels, and ultimately that's what we're trying to do. But we haven't seen any other companies doing exactly what we're doing.
So can you fill in a couple blanks for me? So this is a small molecule?
Small molecule.
As a modality.
And it's passing the blood-brain barrier.
And how are you administering it to these transgenic mice?
That was done through injections, and we are aware of the fact that an oral drug would be ideal in many cases. And so what we're doing at this point, the funding that we're also trying to seek is not only is it for some of this really baseline toxicology, the pharmacokinetics, pharmacodynamics, but is also looking at whether it could be an oral drug as well. So, I mean, we have our team right now with the foundations at Cal State Long Beach. We have team player from Avenir. We're also looking to align with other strategic investors that could help guide the path as well in terms of how we would do that. A consulting team as well that we're looking to bring in that has about 3 team members that have Alzheimer's background as well. So we're really looking to...
Because I'm also reflecting back then on your ASK slide, it is $2 to $5 million to power through the IND enabling studies, which in my world, it leads one to think that you have a development candidate. But if you're still trying to figure out the formulation for your ideal route of administration, then you're actually a little bit farther away from actually kicking off an IND enabling study because you don't have your clinical candidate yet. And again, I'm just giving you this feedback just so that you can kind of sort of reformulate truly, to express truly where your stage of development is at. I think you have some tantalizing pieces of data here. Again, you only have so much time and so many slides to go through with us, but you're also missing the manufacturing cost. Yeah. Because part of your IND enabling study is also creating enough at scale
Yeah.
And then also getting it into that clinical grade. Again, with a small molecule, it's not terrible, but you have to put that into your calculus, into your ask, because otherwise you're going to have to end up fundraising when you're even less than halfway through.
Yeah.
And you don't want to do that.
Yeah. Well, we've been looking at the CMC and the GLP and GMP and all those things. So, we have established funding for that. But again, we're coming from the academic standpoint, and this is why we're looking at alignment with other players, other investors. Originally, when we came out, right off the bat, the intent was to connect with pharma, and we actually spoke with the pharma companies a few years ago, and they said, "Well, why don't you go do some transgenic mouse models and come back to us and let's talk." And we did that, and they said, "Okay, great. Well, where's all the where's all the toxicology?"
I understand the goalpost moving thing can be frustrating, but that's why it actually behooves for you guys to kind of at least plot strategically, especially on that bar graph of like, we need to do this, we need to do this, we need to do this. It takes this much to do this, each segment. And then you leave it to your investor, to your partner, say like, okay, maybe somebody will write you a huge check to get you all the way into first human. And maybe somebody will only do a discrete thing. But if you don't show that level of guidance and knowledge, it becomes a little challenging.
Yeah. Well, we've got the team that we're talking to that has that firsthand experience bringing a few Alzheimer's drugs, at least through clinical trials. And so that's where we're at. But this is the current state where we're at. And we're really, thankful to be involved with this type of a forum and really appreciate it.
Mark, those values you've got over there, it looks like 10 milligrams and 20 milligrams as doses. How potent is this molecule? It strikes me as not that potent.
I didn't quite understand. You said, can you repeat
how
the question?
potent is the molecule, and how much do you think you still need to optimize it? The doses over there look very high.
We'd have to talk to our chief scientist about that, but these are the doses. We tried 10 milligrams, 20 milligrams. It's a small molecule, and it was all done, through, administered shots to the different transgenic mice. And so that's how we did injections. And so I don't think I answered your question, but if I
No, that's fine. I think it goes back to the same point that Tari's making, right, is knowing exactly where your molecule is and how much optimization you're going to need until you add a development candidate.
Yeah.
Would be.
But also, is that mg per kg? It might not be.
Yeah, I think that's just straight.
Well, we're looking at this next round of conducting some of that work and really looking at the pharmacokinetics, pharmacodynamics. Dosing was a big part of that. We have the whole thing mapped out for 2 years, and dosing was a big part of that as well.
And one of the things I highly recommend, I've worked with a lot of companies who've developed drugs in Alzheimer's, and I think when they've started putting a lot of focus on their preclinical data and dose determining data on mouse models, I know we have to start somewhere, but I highly recommend you also look at humanoid models. There are a lot of companies that sell brain models right now that really study CSF, because that's going to tell you, that's going to get you closer to understanding, the efficacy of your drug and also your dose determining efficacy. So I highly recommend you look at some of these humanoid models, especially because now with the failures with other companies, regulatory agencies are not obviously clinical data, but humanoid kind of model.
Yeah. Well, that's great advice. And, ultimately we're following the path here and taking the advice that we can. So, and that's why we're going about it. Just following the journey here.
That's all you can do, right?
Yep. Okay.
Thank you very much.
You.
Great presentation.